At first glance, ctenophores, or comb jellies, look like drifting glass ornaments, translucent, fragile, and almost unreal. But that first impression hides a biological paradox. These animals are soft-bodied marine invertebrates found across the world's oceans, from shallow coastal waters to deeper offshore environments, and they belong to their own major animal group, the phylum Ctenophora. What makes them instantly distinctive is movement. Instead of fins, muscles, or jet propulsion, they swim using 8 rows of coordinated cilia, tiny hair-like structures often called comb rows. Light refracting through these rows creates shimmering rainbow bands, one reason they are often mistaken for decorative jellyfish. Yet ctenophores are not jellyfish, and that distinction matters. They are a separate lineage with different anatomy, different prey capture strategies, and potentially a very different place in animal history. In this explainer, we are not just identifying a strange sea creature. We are tracing how an animal that looks simple on the outside can force scientists to revisit assumptions about predation, body design, and even the earliest branches of complex life. To understand ctenophores, start with scale and structure. Adult forms range from just a few millimeters to around 1.5 meters, an enormous span for a group that is often described casually as gelatinous plankton. They are also the largest animals known to use cilia as their primary means of swimming. Their body is built around mesoglia, a jelly-like internal mass, with a 2-cell-thick outer layer and another lining the internal cavity. That architecture makes them mechanically soft but functionally sophisticated. Their comb rows beat in coordinated waves, allowing controlled movement through water, including directional adjustments and vertical positioning in the water column. Unlike animals that rely on bursts of power, ctenophores exploit continuous micromotion, turning countless tiny ciliary strokes into steady propulsion. This is elegant from an energy perspective and highly tuned to marine fluid dynamics. In effect, the entire organism behaves like a living interface between body design and water behavior, where motion, feeding opportunity, and survival are tightly coupled. Even before we discuss hunting or evolution, the locomotion system alone makes ctenophores a remarkable case study in how nature can scale delicate microscopic mechanics into whole animal performance. One common misunderstanding is to treat ctenophores as a single body type. In reality, the phylum includes strikingly different forms, and those forms reflect different ecological strategies. Cydipids are often egg-shaped and equipped with a pair of retractable tentacles used to capture prey. Platictinids are flatter and generally combless in appearance, an adaptation that aligns with very different lifestyles from free-swimming forms. Baroids, by contrast, are large-mouthed specialists that prey on other ctenophores, showing that even within this group there is direct predator-on-predator pressure. This diversity tells us an important story: gelatinous does not mean primitive in function. Different ctenophore lineages have modified shape, feeding structures, and movement style to occupy distinct niches. Some are built for drifting interception, others for active engulfment, and others for benthic or substrate-associated ways of life. So when we ask what a ctenophore is, the honest answer is not one silhouette but a design space. Their shared core traits define the phylum, but their variation demonstrates evolutionary experimentation at multiple levels, from gross morphology to hunting mechanics. Feeding behavior is where comb jellies become even more compelling. Almost all ctenophores are predators, and their prey range stretches from microscopic larvae and rotifers up to adult small crustaceans. That span indicates opportunistic but structured predation across trophic levels. Rather than relying on stinging cells like cnidarian jellyfish, many ctenophores use adhesive capture strategies through specialized cells on tentacles, then transport prey toward the mouth. Baroids take a different route, using large oral openings to consume other comb jellies directly. In both cases, predation is not random drifting luck, it is built on sensory and mechanical timing. The animal must detect potential prey, align body orientation, and execute capture before turbulence or escape breaks contact. This makes ctenophores active participants in marine food webs, not passive blobs. Their population dynamics can influence plankton communities and, in some contexts, broader ecosystem energy flow. Even rare exceptions sharpen the pattern. Juveniles of two species can live parasitically on salps before transitioning to the predatory behavior seen in adults. That life stage shift reminds us that ecological role is not always fixed across an organism's lifespan, even in groups we tend to oversimplify. Now consider the fossil problem. Ctenophores are soft and gelatinous, exactly the kind of body you would expect to vanish without a trace. Yet fossils interpreted as ctenophore-like forms appear in exceptional preservation sites dating back to the early Cambrian, roughly 525 million years ago. That timing places them near one of the most consequential intervals in the history of life, when many major animal body plans emerge in the record. Fossil interpretation is never trivial, especially for soft-bodied organisms where diagnostic features may be incomplete or altered by preservation bias. But even cautious readings suggest that ctenophore-like animals are ancient, which means their key innovations, including ciliary locomotion at animal scale and specialized predation, have deep roots. This ancient presence adds weight to modern phylogenetic debates, because if ctenophores branched off very early, then traits we see in them might reflect either very old retained features or highly derived adaptations that occurred surprisingly early. In other words, fossils do not simply date their existence, they intensify the question of what ctenophores can tell us about the earliest evolutionary architecture of animals. The most controversial question is simple to ask and difficult to answer: where do ctenophores sit on the animal tree of life? For years, many frameworks placed sponges as the earliest branching animal lineage, with ctenophores splitting later. But several molecular phylogenetic studies challenged that order, proposing the ctenophore sister hypothesis, in which comb jellies diverged before sponges and thus before the rest of animal diversity. If true, this would have major implications for how we interpret early animal evolution, including the origin and loss of complex traits. Why? Because ctenophores possess organized tissues, nervous and muscular systems in distinctive forms, and active predation. If they truly branched first, either these features evolved very early and were later simplified in other lineages, or similar functional systems arose independently in different branches. Both possibilities are profound. They force us to rethink linear narratives of increasing complexity. The debate, then, is not academic hairsplitting. It affects foundational stories about what the first animals were like and how often evolution can invent comparable biological solutions under different constraints. Why has this debate been so hard to settle? A major reason is methodological sensitivity. Deep evolutionary divergences are inferred from modern genetic data using computational models, and those models can be misled by unusual patterns in certain genomes. Some researchers argued that ctenophore genes are so divergent from many other animals that standard algorithms might incorrectly pull them toward the base of the tree, an artifact sometimes linked to long branch attraction effects. Reanalyses have attempted to control for these distortions by changing gene sampling, model assumptions, and taxon coverage. Yet support has shifted depending on dataset design and analytical framework, which is why strong claims are repeatedly revisited. This is a useful lesson in scientific process. Disagreement here is not failure but stress testing. When data are ancient, noisy, and filtered through model assumptions, robust conclusions emerge only after repeated attempts to break them. Ctenophore phylogeny has become a textbook example of how biology and computation interact, and how conclusions in evolutionary genomics must be read with explicit attention to method, not just headline topology. Recent work has tried to move beyond sequence-only arguments by introducing additional lines of genomic evidence, including synteny, the conserved ordering of genes on chromosomes. One notable analysis reported irreversible syntenic changes in the lineage sister to ctenophores, potentially supporting a branching pattern where ctenophores stand apart early relative to all other animals. Whether this fully resolves the controversy is still debated, but it demonstrates an important trend: the field is expanding its evidence base. Instead of asking one dataset to carry the entire burden, researchers are combining sequence evolution, gene order signals, fossil context, and broader comparative biology. This multi-layer approach is especially critical for ancient splits where each signal is partial and potentially confounded. For viewers, the key takeaway is not that one paper ends the argument forever. The key is that the question is being attacked from increasingly independent angles, and converging support across different methods is the standard needed for confidence. Ctenophores remain central precisely because they sit at the fault line where method, data type, and evolutionary interpretation all collide. Stepping back to ecology, comb jellies matter because they are not just phylogenetic puzzles, they are functioning predators in modern oceans. Their feeding can shape plankton communities, alter competition among small consumers, and influence energy transfer through pelagic systems. In some regions and seasons, gelatinous organisms can become locally dominant, and ctenophores can participate in those shifts with consequences for fisheries-relevant food webs. Their transparent bodies and often delicate forms may hide them from casual observation, but ecologically they can be consequential. Their success also illustrates that evolutionary fitness does not require rigid skeletons or high muscular power. Fluid environments reward different engineering principles: low drag, continuous micropropulsion, adhesive capture, and rapid exploitation of prey pulses. By studying ctenophores, marine ecologists gain insight into how soft-bodied predators persist and sometimes thrive in variable ocean conditions. By studying their interactions with other gelatinous groups, including ctenophore-on-ctenophore predation, we also see that even apparently simple trophic layers contain internal complexity and selective pressure. Ctenophores also challenge how we think about complexity itself. Popular science often frames evolution as a ladder, with early animals imagined as uniformly simple and later animals as progressively advanced. Comb jellies disrupt that framing. They combine minimal-looking morphology with sophisticated locomotor coordination, effective predation, and a disputed but potentially very deep evolutionary origin. Depending on which phylogenetic hypothesis is ultimately supported, they either preserve ancient organizational clues or represent early, radical experimentation that diverged from other animal trajectories. In both interpretations, they caution against visual bias. Transparency and softness are not synonyms for biological simplicity. For research, this means investing in overlooked lineages can yield disproportionate insight. For audiences, it means the ocean still contains organisms that rewrite familiar narratives. So the next time you see rainbow bands rippling through a drifting comb jelly, you are not just watching a beautiful marine curiosity. You are looking at an animal group that links biomechanics, ecology, paleontology, and genomics in one unresolved and deeply important evolutionary story. If you found this helpful, please like this video and subscribe for more explainers.